Hi, I'm Fred Turner. I'm from the UK, and this is my project, building a PCR machine. PCR is basically a process for amplifying and copying DNA. The best way to think of it is like a photocopier, but for DNA. It takes a single strand of DNA, which by itself is not very useful, it's very hard to detect, it's a single molecule, and it copies it about 1 billion times in just a few hours to make 1 billion copies of DNA. And once you've got that many copies, you can actually detect that, you can start doing genetic tests, and then it's useful. PCR was invented before, but the problem is the machines cost about £3,000. This makes it really difficult to actually use the machines and to do research and tests with them at home when you're 17, 18 years old. So the only way I could find to actually use this kind of technology was to build one myself, and I built the machine for £250. The PCR process is basically driven by temperatures, so if you it ramps up and it ramps down to different temperatures, so it'll go up to 95 degrees, down to 55, and back up to 72. And each time it goes through the temperatures, the amount of DNA doubles. I also had to develop some other bits of equipment, so I developed a gel electrophoresis box, which allows you to visualise and actually see the DNA after you've done the PCR process. Because DNA is transparent, so you actually need a stain, and then you need what's called a transilluminator to actually see the DNA. And that then allows you to take pictures, such as this one here. This is the first PCR reaction I ever ran, and these two bands here are bits of my DNA that I've amplified. And you can see here, this is my lab at home. So you've got the PCR machine here, electrophoresis, and then there's other bits of equipment which I actually had to buy second hand, such as a centrifuge, which isn't very safe to try and make yourself. That's it. Thank you for watching.